So prepare your pen and paper or pencil if you prefer. We're gonna write down certain things. And I wanna make it relatively general so everybody can understand, hopefully, even without really deep chemistry knowledge. However, the main kind of topic is actually chemistry. Oops, a little already. So first of all, a few words about my funding. Just want to thank Alexander von Humboldt Foundation, and in particular Sofia Kovalevskaya Award, because of which I'm here in Germany. And because of this funding, I'm able to speak with you about the research we are doing in Helmholtz Institute Mainz and Johannes Gutenberg University Mainz. So first, a few words about this topic. We are doing NMR. However, the world is facing global challenges, and one of them is energy, clean energy. And there is a portfolio of technologies being developed which actually use hydrogen and try to store energy in the form of hydrogen. These are called smart technologies. Actually, energy can come from different sources, and hydrogen is a very nice, it's called green platform for storing this energy, and many European countries, including Germany, are really heavily participating in this direction. And I'm very happy to be in Germany as well. But I'm also happy that hydrogen can be used for completely different purposes other than energy storage. And this is enhancements of nuclear signals in magnetic resonance imaging and nuclear magnetic resonance. And for that, we need to understand the very, very basic quantum mechanical concept, which is entanglement, and the fact that hydrogen actually consists of the mixture. Normal hydrogen molecule consists of the mixture of two different nuclear spin states. First, it's called orthohydrogen, a state with a total nuclear spin 1. So we can call it a triplet state. You have alpha alpha, 1 square root alpha beta plus beta alpha, and beta beta. And the second state is called parahydrogen, which is a state with a total nuclear spin 0. So it turns out that you can relatively easily obtain parahydrogen by simply cooling hydrogen gas. So why does it work like that? It turns out that hydrogen, it's a very small system, it's a small molecule, and nuclear spin levels and nuclear spin states are connected to rotational energy states. And because moment of inertia of hydrogen molecule is relatively small, gap between energy levels, so if you plot energy and if you plot energy levels of rotational energy states, you may see that rotational state with the 0 rotation in the first, and then so on and so forth, this gap is relatively large. If you really use h omega 0 equal to kt type of approximation, you can estimate this gap of about 80 Kelvin. So what does it mean? It means that if you were able to reach equilibrium, basically equilibrium of rotational energy levels at 80 Kelvin, so which is approximately temperature of liquid nitrogen, you would populate equally state with rotational energy level 1 and state with J equal to 0. So and that's what we are doing in our research very often. We basically use liquid nitrogen parahydrogen generators. We simply flow hydrogen gas, which is normal hydrogen gas at room temperature, 3:1 ratio of ortho and para isomers. You can flow, actually the wrong way, this way, into the dewar filled with liquid nitrogen, and then you have a spiral tube, and at the end we normally put a catalyst of ortho-para conversion, and we very often use some paramagnetic materials like iron oxide, usually this form, iron OOH, and then the gas, as it flows back to room temperature, if we remove catalyst of ortho-para conversion, it can stay in this para state. So first thing to understand, nuclear spin energy levels and rotational energy levels are connected in hydrogen molecule, and by employing rotational energy levels, we can actually effectively cool down nuclear spin states and obtain all hydrogen molecules in the para state if we really go very deep. For example, if we go to the temperature of liquid helium, and already at above 25 Kelvin, we can have very, very good hydrogen almost 100% parahydrogen enrichment. So that's the beauty of this approach. You can obtain parahydrogen relatively easily. You can relatively easily obtain this enrichment. And second, it's very easy. You can store this parahydrogen in a tank, and if this container is clean, it can be stored for days and weeks without back conversion to the ortho isomer if you don't have a catalyst of ortho para conversion. So that's a very unique initial state, so to speak. We can initialize our state chemically in a very cold nuclear spin state. So, and this idea to use parahydrogen in NMR appeared not immediately. So parahydrogen was known in literature, in research, for reasons of fuel for rockets. Basically, it was clear that you can have this ortho-para conversion, and if you don't use a catalyst of ortho-para conversion, if you just quickly cool down your hydrogen gas and don't worry about converting ortho-para hydrogen into parahydrogen, you'll have excess energy, which eventually can start heating your gas and can evaporate it, create some pressure, kind of overflow, and basically it's dangerous. You need to convert all your hydrogen fully into lowest quantum state, and that was known for a while in rocket fuel community, but applications for NMR started from this publication in 2087 by Bowers and Weidekam, who introduced so-called Pasadena effect. So they call it Pasadena. Parahydrogen and synthesis allow dramatically enhanced nuclear alignment. And that's how it was introduced in the literature first as a thought experiment, and then later in this publication, it was demonstrated that you can indeed obtain enhanced and very specific multiplets in NMR, which you normally wouldn't obtain with a positive and negative enhanced lines. So we call it multiplets, anti-phase multiplets, and that's a signature of using the spin order from parahydrogen. So let me see if there's, by the way, if there are questions, feel free to ask right now. I can answer them right away. All right, let's see. Basically, let's go straight to the point. So applications of parahydrogen are actually abundant, not only for chemistry. So since the beginning, since the 1980s and 90s, a lot of chemists started using this effect, because indeed you can employ this hyperpolarization by parahydrogen for studying reaction intermediates in catalysis and catalytic reactions. However, another application is MRI. Imagine the following situation. You can actually attach parahydrogen to a molecule which will eventually become a metabolite in your body. Metabolite which is totally normal and normally present in living organisms. So by injecting this metabolite, hyperpolarized metabolite, you can see where in the body this chemistry is going, and second, you can see actually chemistry that is happening. So it's very, very interesting, in my opinion, direction, and it was understood from the very beginning of parahydrogen-induced polarization phenomena from Pasadena that it can potentially be used in MRI. And I think these days we are very close to actual applications, even in humans, not to mention animals, of parahydrogen-based research. And I just want to point out that it's somewhat similar to what's happening in positron emission tomography, where people inject radioactive tracer and they see indeed using gamma ray imaging, basically, or by imaging gamma rays emitted due to annihilation of this radioactive isotope, you can see where exactly in the body molecule is located. And it's very useful for cancer diagnosis and treatment. However, it's radioactive and it still does not provide chemical information. So if a radioisotope is modified chemically, if the molecule, if the atom, radioactive atom is transferred from one molecule to another, you're not going to see it. You still see location of this radioactive molecule, radioactive atom, but you don't know what chemistry happened. However, with MRI, if you were able to polarize your signals to a large extent, you would be able to see not only where molecules are going, but also would be able to see chemistry. All right, I see a question by Karina. Quick question: are you quantifying the amount of ortho to para conversion? And if so, how? All right, very good question. So normally, what do we do in NMR? You actually can see directly ortho-para ratio by using Raman spectroscopy. And that is done by some groups. So you actually will see peaks associated with ortho and para hydrogen at different rotational levels. And you can simply, by integrating them, you can actually analyze your ortho-para ratio. In fact, we have a simpler solution. We are just looking at NMR directly. So we have hydrogen gas flowing, let's say, after a generator, and it can go in an NMR tube, and we can have outflow, and we also have, let's say, some pressure valves and safety valves, such that we can control pressure in the tube. And we just looking directly, this is, let's say, our RF region in an NMR tube, by NMR at the tube through which we are flowing hydrogen gas. And if we have, let's say, normal hydrogen gas, right? And I recommend doing this at elevated pressure because T1 and T2 of hydrogen grow up when pressure goes up. So and it will help you actually to see narrower line in your spectrum, but also you can just see that 3-to-1 ratio of hydrogen, which is normal hydrogen, will give you, let's say, signal S, let's say call it S1, and then you want to find out what's the ratio, what's the X ortho-para ratio that you have in your mixture after parahydrogen generator, and you have a signal S2, and then you can, by doing very simple proportion, you can estimate, for example, how much by looking at NMR signal, because NMR is given always by your ortho-hydrogen. So by knowing this fact, you can actually very well estimate your fraction of parahydrogen. And if you have 100% parahydrogen, indeed, you shouldn't see any signal, even from a very well pressurized NMR tube. So that's what normally we do in our research. All right, let's see. All right, how does it work? How does this NMR enhancement works? And let's start from the very basic. First of all, the whole portfolio of parahydrogen-based effects is called parahydrogen-induced polarization. And I think that's how it should be called. It was discovered by Bowers and Weidekamp. However, since then, many groups started developing, and I apologize immediately for maybe some people who I will not be able to mention due to the lack of time during this talk, but there's many groups and research directions evolved since these first initial experiments. But generally, all of this parahydrogen phenomena called parahydrogen-induced polarization, or PHIP. So how does it work? If you have parahydrogen itself, just two spins at low field or at some field, you have an energy gap determined by your actually J coupling, or even in case of free hydrogen gas in a gas phase, it's actually much larger than J coupling. This gap between ortho and para states, as I said, is 80 Kelvin. It's a rotational transition between 0 and first rotational quantum level. However, if you're talking about hydrogen and consider only nuclear spin levels, at high field, you're obviously going to split your triplet levels according to your field at which you do experiment. But let's say that you prepared and you enriched your hydrogen with para isomer to 100%. So all population sits in this alpha beta minus beta alpha level. And by the way, I'm going to use this terminology when I put a ball on the level. It means it's a population of this level is overpopulated. So, and you can think of NMR transitions, observable transitions, are being transitions between ortho states. So the signal actually of ortho hydrogen is not really, it's proportional to the concentration of your ortho hydrogen in your mixture, as well as it's still proportional to the difference between populations. And here this difference will be very small. It's proportional to the normal Boltzmann distribution in the high field. So now imagine that these two hydrogen atoms now end up suddenly, you have a chemical reaction at high field. They end up in a different chemical environment. So the first hydrogen have one chemical shift, second hydrogens have a different chemical shift. So now a good basis to describe your molecule is a Zeeman basis, especially if chemical shift difference between these two spins is much larger than J coupling between them, which is a typical case in actually organic molecules when you have hydrogenation reactions, and that's what we're talking about, hydrogenation reactions. And it's clear that from alpha beta minus beta alpha state, you're going to end up, it's like a projection. You project and you can end up either in beta alpha or alpha beta states. And by the way, the gap here is a little bit exaggerated. In reality, if you're really talking about high field, like fields above 1 Tesla, so the gap between alpha beta, let's say in alpha alpha, is much larger than the gap between alpha beta beta alpha. But here, just forgive me for this oversimplification and exaggeration. So in reality, what you see, because of this picture, you're going to see transitions both up here, as well as emission. We call it emission because the lines are enhanced down. So and that's what you observe if you do hydrogenation reaction at high field. And this is called Pasadena effect. This is exactly what was discovered and described by Bausch and Weitekamp. Few years later, same authors, Weitekamp, Pravica, and Bausch, they discovered another effect, which is called Altadena. And it's coming from, actually, I forgot the exact terminology, adiabatic longitudinal transfer, and that enables Altadena, net alignment, something like that. Actually, I forgot the exact terminology, but the idea is the following. You do hydrogenation reaction and forget that you have other spins in the molecule. Let's say you have just still no protons in the final molecule, you just attach parahydrogen to some substrate, and two atoms have difference in chemical shift. However, you still reside at low field. Low field means that chemical shift difference between these two protons is still smaller than the J coupling, and let's say gamma and B is 0. That's your field. So difference in resonance frequencies is smaller than J coupling between them. And that's a condition of low field for protons generally in simple situations when you're not talking about some heteronuclear spins involved. So it means you're still gonna stay in the same basis. So the same singlet triplet basis is still good basis for describing your spin system. So singlet state will stay unperturbed. However, then if you adiabatically, meaning relatively slowly, increasing the field, so after performing hydrogenation reactions, you're going to overpopulate only one state. It's going to be either alpha beta or beta alpha, depending on the sign of J coupling with respect to the difference in chemical shifts. And you're going to have a situation where basically, let's say, transitions are only possible between these levels. And in the spectrum, schematically, you're going to have one spin up, another spin down. Unlike the previous situation in Pasadena case, where you had up, down, 2 pips. So here you can actually say that one spin is polarized up and another spin in an ensemble sense, meaning that in every single molecule, left spin is up, let's say, and right spin is down. So, and that's called Altadena. And as I said, for many years, Altadena and Pasadena have been used in organometallic chemistry and in general in catalysis to study reaction intermediates, things which would show up in very, very interesting regions, for example, -16, -10, -20 ppm. And usually those are hydrogens attached to metal. And by the way, this reaction, and I haven't mentioned it, haven't focused yet, it happens, it requires a catalyst. So hydrogen has to come to the product molecule in a pairwise manner, which means two hydrogen atoms from the same molecule should retain its nuclear spin memory between each other. And that's what we call pairwise hydrogen addition. It's usually realized using homogeneous catalysts. And a few words about homogeneous catalysis in the next slide. So today, I'm gonna briefly touch upon and we already discussed PHIP, parahydrogen-induced polarization, but there is also a sister variant, actually its extension, which is called Saber, abbreviation for signal amplification by reversible exchange. So it's a very interesting phenomenon. Here, instead of reaction, one-way attachment of hydrogen to the product molecule, you can actually have a reversible association and dissociation, so exchange process, and your final product that you want to polarize doesn't actually be chemically modified in Saber. So it allows to polarize different classes of materials. Usually it's done using different chemicals. And here, that's what we mean by catalysis. So catalyst is a compound that allows a transfer of spin order from parahydrogen to the substrate. In both cases, you need these compounds. So what these compounds could be? And in general, in chemistry, there's a terminology of homogeneous and heterogeneous catalysis. What does it mean? Homogeneous catalysts are usually complexes of platinum group metals. It can be rhodium, iridium, platinum, palladium, ruthenium, and actually a lot of metals from the periodic table. And they're covered in organic ligands. Usually these ligands are necessary to, first of all, dissolve your metal center in the solvent that you are using for your reaction. And second, to stabilize the complex. And by tuning properties of these organic ligands, you can actually modify a lot properties of your chemistry, exchange rates, for example, attachment ability of hydrogen to attach and dissociate, ability of your substrate to dissociate if you want that. So it's a whole field, very interesting field of organometallic chemistry. And then you have heterogeneous catalysis. These are typically reactions and catalysts being metal particles, for example, clusters of platinum or rhodium on the surface of usually inert solid support. And as supports, very often there is a silica or a silicon oxide, titania, alumina, and actually even sometimes carbon and other supports can be used. So all of them were shown to demonstrate that indeed sometimes in certain reactions, a small portion of hydrogen molecules, when the hydrogenation happens, still can attach to the product in a pairwise manner. However, if we are really talking about large polarization values for biomedical applications, and by large, I mean routinely more than 5%, let's say, carbon final polarization as a threshold, we would be currently still talking about homogeneous catalysts simply because so far there have not been good heterogeneous system demonstrated for this application. It doesn't mean it's impossible with heterogeneous systems, and maybe one day we will see a very nice heterogeneous system where we simply bubble the hydrogen gas through the solution of water and our chemical inside, which we want to hyperpolarize, and some solid particles, which are actually not undergoing leaching, they're staying on the surface, staying in the solid state, can be used for that. So that's one of the, I think, really nice directions of research. However, in most cases, currently, we're still talking about homogeneous catalysis. So if you have questions, again, I'm very open right now to have a small break before we move into details of Saber.
Please post your questions onto the Q&A window. You should be able to access that directly from your Zoom interface.
All right.
Yeah, sorry, maybe before the audience posts some questions, Danila, I also had a question. Like initially, we're talking about the advantage of potentially improving the sensitivity of a metabolite and then using that in, in MRI. For something like that, how long do you think the polarization would last when enhanced by this method? Yeah,
so basically, we need to distinguish, so what type of nuclei we want to polarize. Right. So far, I only focused on hydrogens themselves, protons, 1H. And we focused so far on the mechanism of breaking this symmetry of parahydrogen molecule to be able to manipulate this spin order. But once, for example, you have a molecule, let's say here, and you have a nucleus, for example, C13 or N15 in the molecule. As you have broken the symmetry of parahydrogen in this molecule, now you can use polarization transfer sequences to transfer polarization directly, basically from singlet state in your hydrogens to carbons. So in terms of lifetime, usually if you store polarizations, let's do you carry down the reaction in hydrogen on the hydrogen. So lifetime can be seconds, maybe in unique situations. On hydrogen itself, it can be more than a minute. Usually it's not T1, it's some sort of interesting singlet states like the ones shown here. So if we retain this symmetry of parahydrogen in the product molecule, in certain situations, you can end up in a quite long-lived state, but usually it is seconds. And in order to preserve polarization, in order to really use it for something useful, most of the time you want to immediately, once you produce parahydrogen polarization, transfer it to carbon or to nitrogen to allow it to survive for much longer time, and then inject, for example, and observe metabolism directly on this heteronuclei. Or alternatively, you can still bring it back for detection on protons, because for sensitivity reasons it can be advantageous, but for storing reasons, I think it's most reasonable to store on heteronuclei such as carbon, nitrogen, or some more complicated spin states.
Okay, thank you. So I would like to just encourage the audience to continue to just post your questions on the Q&A window, as we are likely to take more breaks during the talk, and I'm sure Danila would be happy to answer them.
Mm-hmm. All right, let's continue. So basically, here is the general schematics of Saber, which is signal amplification by reversible exchange. So in here, you have also catalyst, again, magnetization transfer catalyst. It's not a real chemical catalyst. It doesn't really catalyze brute chemical transformation, but it endows and it enables ligation of both parahydrogen or hydrogen molecule and a substrate of interest in a reversible manner. And as a clear example and practical example of this, let's look at the reaction with pyridine on this iridium organometallic complex with the special ligand I-mesh. Although it's an N-heterocyclic carbene ligand, there can be actually many different ligands found in literature, and even simpler structures such as Crabtree catalysts can be found, and you can some of them are commercially available, some of them you have to synthesize. But the idea that these complexes exist, and complexes in which there is a reversible interaction of this ligand on a time scale of, let's say, inverse 10, about 10 inverse seconds. So it's actually milliseconds, tens of milliseconds lifetime of this complex before it dissociates, a new hydrogen molecule can come in. So what's the orta, meta, and para protons of pyridine to extremely large values by simply bubbling parahydrogen through the solution for a few seconds. So actually this is amazing, and you can polarize pyridine to a very large extent. So to understand how exactly it works, let's look at the simplest spin system which is suitable for the purpose, which is 3-spin system. And I want to say right away, this is an oversimplified description of the Saber process. In fact, if you want to look at the details, you can go to this review. I think we summarized a lot of polarization transfer mechanisms important for Saber, as well as chemistry. And I want to point out that there's more developments in the field. Right now, the description which I'm going to be using is shown to be actually not always applicable, in the sense that you can sometimes achieve polarization levels even in regions which do not are not explained by the picture which I'm going to provide. However, this picture is very basic, and it's very simple for beginners to understand how do you obtain polarization in Saber method. So imagine that you have a complex in which you have 3 spins, and 2 of them come from your parahydrogen, and a 3rd spin is from your substrate molecule. In this particular case, I put it on nitrogen side. However, it can be even proton in the spin system or any other heteronucleus. Sometimes this 3-spin system is simple but good enough to explain the underlying dynamics. And you can show if you have a certain suitable interactions between the spins. In particular, you need to have J coupling in the complex, and it should be inequivalent J coupling, such that 2 hydrogens originating from the same parahydrogen molecule should have a different interaction with the spin that you want to hyperpolarize. And in that case, if you plot energy level diagram of this system as a function of field, you'll see this interesting situation, and we call it lux, or level anti-crossing. So this is a situation when some levels tend to cross, but they anti-cross, and there is a gap between these levels. And for this three-spin system described here, it's actually very convenient to use singlet triplet bases, meaning that we describe the state of a hydrogen spin pair as singlet or triplet. So the first, you have triplet minus beta, for example, means beta, beta, beta. Singlet beta, it means the singlet state of hydrogen, and beta is a state of your third spin, in this case, exemplified by nitrogen. So what's the key idea? The key idea that by saturating solution with parahydrogen, by bubbling hydrogen gas through your mixture of your chemicals, you actually overpopulate singlet states in this system. So among all of these levels, level singlet alpha and singlet beta will be overpopulated because your third spin is not polarized. You can say in the first approximation, alpha and beta are equal in energy. So, and then by saturating with parahydrogen, you can actually see that in this level anti-crossing, and that's a useful picture. And the reason why we like this level anti-crossings, you can start polarization transfer between levels associated with level anti-crossings if you come and do your bubbling of parahydrogen at that field. So indeed, you notice that singlet alpha can be transferred into triplet plus beta. Note that total projection is conserved. So in total, energy is conserved. However, singlet beta is staying as singlet beta. So from initially almost equal population of alpha and beta, meaning zero polarization of your substrate, now you have negative polarization of your substrate, everything is better. So in ideal case, actually, from 1 mole of parahydrogen molecules, you can in principle obtain 1 mole of hyperpolarized material. In reality, it's always more complex. You have relaxation processes, you have other dissipation processes in the system, and you obtain lower polarization levels. However, indeed, we are at the level when we can obtain levels of 10, 20, 30%, sometimes routinely inside our benchtop NMR spectrometers. So I think we are really, with this technology, we are very easily talking about very large polarization values on the order of unity. So now I switch gears to this new technology extension of the Saber and PHIP technology, which is called Saber relay and PHIP relay. And I will have a few more slides explaining PHIP relay. So what's the idea? It turns out that, okay, you can transfer, we know, and there's a lot of research, we know that we can polarize a wide class of molecules, but some molecules still cannot bind to this magical, wonderful iridium catalyst, or sometimes you cannot hydrogenate some molecules directly with parahydrogen. So what can you do? And it turns out that it is possible to relay, to transfer further polarization from the molecule that you polarize by Saber or PHIP to other molecules, usually employing further reactions. And it was also demonstrated by Professor Duckett and his group, an example of Saber relay. So when you polarize things like NH3, ammonia in solution, and then ammonia is known, especially if you choose a proper solvent, usually it's aprotic polar solvent. It should be a solvent which can dissolve polar molecules as well as support hydrogen exchange, yet you shouldn't have that much hydrogen exchanging freely like water. And in that case, you can exchange these protons with other substances like alcohols, acids, amides, and do still observe polarization if your initial polarization was high. And it's indeed demonstrated. So in here, I just show an example of our own work, and it's done by my student, Eric van Dijk, where we hyperpolarized and we optimized hyperpolarization of methanol and ethanol by using benzylamine as a polarization transfer agent. And for this work, we used benchtop NMR spectrometer, which is quite convenient. You can do all experiments and optimizations routinely. And you can see that indeed, as a function of catalyst concentration, as a function of a substrate ratio, we have very non-trivial dynamics where we don't, I wouldn't say that we understand everything in the final level of detail. However, we know that we can find conditions, sometimes experimentally, where we can polarize certain chemical groups quite efficiently. So the idea here is, again, that instead of NH3, we use benzylamine. It also have NH2. It also can bind to the catalyst, to iridium complex, and polarization can transfer from parahydrogen to hydrogen atoms in NH2 group. And then this NH2 group is transferred, exchanged with OH group in methanol. And because everything is happening at low field already, and the exchange is controlled by concentrations and the choice of solvent, we can find conditions where exchanges slower and low enough when polarization transfer to other protons can naturally happen due to J-coupling network in the molecule. And this is a type of experiment that you can do with optimized samples. And we take vodka. It's really store-bought solution, store-bought sample. And we take aliquot of that sample into our solution, which we already know optimized. So we optimize for catalyst concentration, we optimize for concentration of benzylamine. And you see an example where on top is a spectrum at a thermal equilibrium at 1 Tesla. So it's basically benchtop NMR spectrometer with 1 Tesla. You can, in principle, get a better sensitivity than here for the solution at 1 Tesla if you use higher receiver gain. We deliberately put the lowest receiver gain because we want to compare apples to apples, exactly in the same conditions with the hyperpolarized spectrum. And that's what we see in comparison when we do about 20 seconds of parahydrogen bubbling. So you see the same solvent peak is dichloromethane, and actually even solvent peak is enhanced compared to thermal solvent peak at 1 Tesla. And the nature of this, I can talk a little bit more in detail. It's a cross relaxation of hyperpolarized material. But what's more important, we see very strong enhancement of all other peaks in our chemistry in total, not only benzylamine, our polarization transfer agent, we also see hyperpolarization of ethanol, very well visible, and remarkable that it's actually from this external sample which we took extract from. So I think the analytical potential of this technique is quite amazing, and I don't think it's fully explored actually. And I invite many more groups to explore it. If you have a benchtop NMR spectrometer, if you have these simple chemicals, you don't really need to use deuterated solvents. You can work in protonated solvents now. Actually, there's also techniques for solvent suppression. So I think we're entering very interesting time when a lot of research can be done in a straightforward manner and with relatively small investment, so to speak. And this is more advanced kind of setup that we have in our lab. So we have, let me see if I can show the video. So we have a robotized setup which allows transferring our sample with all the tubing for parahydrogen supply. It's actually under some elevated pressure, about 3 to 5 bars. We can transfer parahydrogen and really automate acquisition of our spectra. So we can do parahydrogen bubbling at different fields, at Earth field, at some millitesla field, and sometimes we do it in microtesla field. So where actually we shield our environment, our sample from Earth field, and we do it with the help of mu metal magnetic shielding, so which was demonstrated here. So the advantage of this, that at high field, we still have really nice sensitivity of NMR detection, and we can address spins selectively. However, at zero field, we can use the fact that spins are talking to each other. So energy levels are so to speak, mixed at zero field, and we can use it for both polarization transfer, which we're using, and for generation of polarization with Saber in particular, as well as for many more interesting experiments, which we started doing. I don't have much time, unfortunately, to explain exactly what we're doing, but we are talking about quantum simulations and indeed taking advantage of strong coupling between the spins, and it is a so-called Heisenberg Hamiltonians. Only nearest neighbor interactions are dominating due to the fact that J-couplings are very effectively they're transferred by electrons through chemical bond. So in a chain of spins in a molecule, you actually have really nice Hamiltonians for which there's a lot of solutions in literature. So you really can start thinking about maybe not quantum computing, but at least some very interesting quantum simulations using nuclear spins in solution in liquid state. And this direction was actually known for some time, but I think only now we are really coming to realization of how we can initialize our spin states with really large polarization values on the order of unity, which is very similar to basically creating pure states. So in a sense, when you have large polarization in the ensemble of molecules, it's very similar to working with individual molecules. So, and another direction that we do with this hyperpolarization methodology, we're using different sensors. Instead of using normal NMR, which is benchtop NMR spectrometer or high field NMR spectrometer, we can use optical magnetometers. So these are devices which measure field directly, and you can use atomic magnetometers. You can try to use, there's work published on using very new NV centers in diamonds. There is also a magnetic resistive sensor. So there's actually many different types of devices which can measure magnetic field, and you can start using them. As demonstrated here, for example, on this slide, we hyperpolarize methanol, as I already showed previously with Saber relay. In methanol, in C13 methanol, by simply bubbling parahydrogen gas at low field, and we can detect characteristic frequencies and characteristic spectra at zero field. So this is called J spectroscopy. This is something new compared to high field spectroscopy. It's still NMR, so we measure interactions between the spins, but we're not using chemical shift to discriminate, to distinguish molecules. We are actually using internal spin interactions such as J couplings themselves to distinguish different molecules. And also depending on spin topology, depending on how many spins you have, actually, I don't know, yeah, I think I haven't put an extra slide here. So for ethanol, for methanol, we have different topology between spins. Different number of spins will result in different manifestations of the J spectra. And you can really polarize different molecules which are possessing these exchangeable protons by Saber relay. And then you can study them using this magnetometer. So you don't really need even to buy a high field NMR spectrometer. In reality, we cannot still easily buy NMR spectrometers based on these magnetometers, but that's what we are working here in our lab in Mainz. We're trying to work more and more and develop this methodology of zero field NMR detection, or ZULF NMR, zero to ultra low field nuclear magnetic resonance. And this is types of experiments that you can do, not only observe characteristic J spectra of molecules, you can also monitor their relaxation. If your setup is optimized and robotized, you can do a lot of different experiments. And as I said, in parahydrogen-induced polarization, you can use Saber, you can use PHIP, and you can relay this hyperpolarization further by using third reactions. So, and a previous example of methanol was example of using Saber. Here I want to show a few slides of using PHIP. And it was introduced in literature as PHIP-X, where X stands for exchange. And actually, it was introduced quite recently by our colleagues, Professor Hoviner. And I think personally, it would be great to kind of not invent too many terms, but rather use somewhat unified terminology. So I would call this technique, say, PHIP relay, in analogy with Saber relay, because the mechanism, the idea is very similar. Instead of Saber, you use PHIP, parahydrogen-induced polarization reaction, and you relay by chemical exchange your exchangeable protons to further molecules which you want to polarize. And this is work done by Dr. Shema Alchik, who is now in Frankfurt, a medical center. We used our robotic arm setup, which you saw a few slides previously. And also we did parahydrogen bubbling at low field, and then transferred robotically our sample in the benchtop and did polarization transfer, if necessary, to different nuclei, for example, N15. So polarization transfer can be done using inept sequence. It's one of the actually very simple polarization transfer schemes and it's very well known. There's a lot of books and tutorials about how it works. But the idea of the chemistry is the following. So first you have this molecule which can attach hydrogen, so it contains triple bond. And here we use a rhodium catalyst, very famous catalyst used in majority of actually parahydrogen-induced polarization reactions. And in this case, we didn't optimize for solvent yet deeply. However, as I say, there's a lot of techniques currently available on how you can further clean your solution if you're talking about biomedical application of your hyperpolarization. After preparation of your target hyperpolarization, you can talk about phase transitions such as precipitation of your sample or degassing of your sample or adding some extra solvents to solidify some of the materials and leave some of other chemicals intact. Then you can clean the solution if your lifetime of polarization in a solid state allows. So I think it's not a real problem right now to do and obtain eventually clean parahydrogen-based contrast agents. The main technique, the challenge is still obtaining large polarization on chemicals of interest. And let me show you and focus specifically on one chemical, which is urea. I think urea, both N15 labeled urea and C13 and N15 labeled urea are very interesting. They're used in MRI as well. And there's a lot of discussion on whether we can use them as replacement for gadolinium, actually, because of some toxicity associated with the later chemicals, which has been discovered in recent years. So actually, the idea that urea itself is relatively inert and can be injected in large concentrations and can be used as a tracer. It basically, you just observe where the blood is going. So you can do angiography, you can do perfusion experiments, and there's a lot of interest in polarizing urea to large polarization values, in particular on carbon, but I think also N15 is quite interesting nucleus to investigate. So what we do here, we use this triple bond, which is called propargyl alcohol, and hydrogenate it in acetone with the rhodium catalyst. And we already have all our chemistry together in the NMR tube. So we have our urea, we have our propargyl alcohol, we have our catalyst, we bubble parahydrogen at a low field of about few millitesla. And we actually tested, it's quite broad condition in which you can do this reaction, it still will work. It's pretty much very uniform within the range from 1 to 100 millitesla. So the idea that upon hydrogenation, you have this coupled nuclear spin system and polarization will distribute between spins, including spins inside this of these protons in OH group. And then chemically, OH group is exchanging with NH2 groups, which are present in urea. Actually, they're present in many more different chemical motifs. You can also do polarization of amino acids and sugars and other compounds. So, and what we do in order to distinguish and see efficiently polarization on heteronuclei, after doing this reaction by bubbling parahydrogen at low magnetic field, we transfer the sample robotically to our NMR spectrometer and we do a sequence. And after doing sequence, in particular refocused inept, we can get enhancement on this order. So this signal in gold, you see it's a signal of a hyperpolarized urea at a concentration of about 50 millimolar. No, it's 100 millimolar, yeah, what's written. Compared to this really small, tiny green signal enhanced by the factor of 20 over thermally polarized urea, a very concentrated solution in the same magnetic field. So we're talking here about more than 1% polarization on nitrogen-15 obtained by polarization transfer at 1 Tesla via pulse sequence. So we also, as I said, worked on other molecules such as amino acid lysine. We show that we can polarize ammonia using this technique. We can polarize benzamide. And actually, we haven't investigated many more different motifs. I believe this technique is quite versatile and universal. We also polarize C13 in glucose. And you see that by observing C13 NMR spectra and doing polarization transfer at 1.4 Tesla in a benchtop C13 NMR spectrometer. However, here we haven't achieved 1% polarization yet. I think it probably is still possible. You just need to really do optimization. So my main message here is that we are entering the era of spin exchange chemistry, parahydrogen-based exchange chemistry, where it's known that we can obtain in principle large polarization values. The question is optimization. Question is how we save this polarization and store it in type of molecules that we're really interested in, how we limit and minimize relaxation losses and unknown and unwanted byproducts in the chemistry side. So I think it's very exciting personally to me, and I hope it's exciting to you to see that we actually have a new technology which is, I think, entering now even the market of hyperpolarization. I've heard about companies which are now preparing and selling devices, even in clinics for clinical market and potentially for preclinical market, which are capable of producing these hyperpolarized agents, which are clean from catalysts. So there's a technique and technology there to clean it, the solution from these unwanted chemicals, and produce polarization on a level of more than 30%, which is useful for biomedical applications. So I think it's very, very exciting. I don't think I have time to explain a lot about polarization transfer mechanisms unless there's going to be extra question. And I just want to show the latest type of spectroscopy that we're routinely now doing in the benchtop NMR, which is a activation chemistry of our iridium catalyst, but you actually can do activation chemistry with different types of catalysts, with rhodium, ruthenium, and you see build up and decay as you do bubbling directly in the spectrometer with every, let's say, 10 seconds, sometimes every 2 minutes, depending on what you type of kinetics you're looking at. And you can see that some chemicals go. So you have, we understand partially in some situations what's going on. In some situations, I tell you honestly, we still don't understand. We see that, for example, even here, some intermediate disappeared, then you had nothing in the proton NMR spectrum for some time, and then another intermediate and catalyst complex starts showing up. So we really entering in the scene of these intermediates observed by benchtop NMR spectroscopy, which we don't fully understand. I think it's exciting time for everyone who is interested in spin chemistry and parahydrogen-based chemistry in particular. And just to show maybe very quickly, also one of the latest results, we observe N15 signals at natural abundance from also chemical from the family of pyridine groups, basically chemicals. And we see different complexes, different structures associated with complexes in nitrogen 15 spectrum at 1 Tesla. So once again, some of these peaks we can identify that. So this is a free substrate, and we see some bound substrate species to different types of complexes. So it's very exciting. We don't even fully understand, but the main message is that now we are able to do this chemistry directly at 1 Tesla, something I think even recently has not been even conceivable because of sensitivity limitations. So with this, I want to finish with the quote of Alex Pines, my postdoctoral advisor, who said that indeed parahydrogen has been known as a technique for a while, but now with the latest advancements in physics and chemistry and engineering, we awakened the sleeping giant. I think this chemistry-based hyperpolarization approach is relatively straightforward. It requires a lot of optimization. It's maybe not very general for any type of chemistry, but it's very powerful. And if you optimize, you can, I think, polarize very different chemical motifs for different applications. So with this, I would like to thank my collaborators with whom I'm working now and worked historically. And I would like to thank you for your attention. Feel free to ask more questions.
Yeah, thank you so much for that excellent talk, Danila. I think this has been one of the best tutorials that I've personally attended in recent times, and
thank you.
We are all thankful to you for accepting our invitation to come and speak. So I encourage the audience to post your questions on the Q&A window, and in the meantime, as the audience is typing in their questions, I did have a few questions myself. So I was curious if you could share some thoughts on what are the challenges with applying some of these techniques, especially the Saber relay method that you were showing and doing all this cool benchtop NMR spectroscopy, also at high field? Is that something that you have tried?
I mean, that's
yes.
actually, that's a funny thing from my personal experience. It's actually working in benchtop becomes more practically useful than even working at high field. So for example, at Berkeley, I had access to 800 megahertz instrument, but in practically it turned out that it's really actually challenging to do experiments. So you have this very big device, two-floor device, and you can bring your parahydrogen system. You need to have long lines to move it, to have your magnetic valves and other components below the 5 Gauss line. So you have to control it very well, and you basically inhomogeneities introduced by parahydrogen bubbling itself at high field can lead to basically effects which will affect your, for example, pulse sequence. If you want to do pulse sequences at high field and you want to be very specific in some selective pulses, for example, it becomes just challenging for very simple practical reasons to work at high field. And in terms of our research, what we're usually interested in is large polarizations, right? We are interested in just sometimes observing the phenomenon itself rather than maybe employing the chemical shift distribution, which is available at high field. And I found that it's very, very nice to work at benchtop, in benchtop spectrometers. It's very simple. Certain things you observe just because it's faster, you can do transfer much faster. Let's say manual transfer from Earth field to benchtop compared to manual transfer from Earth to 800 megahertz, right? So only for these reasons. Otherwise, there's no fundamental, of course, reasons why you cannot use high field anymore.
Got it. I was also curious. I think they're still waiting for questions. I was also curious about. So you showed your robotic arm where you're bubbling at these low fields about few millitesla, and then you're transferring to your benchtop NMR. Can you do this? Like, is this NMR signal that you obtain like quite reproducible to do some complex experiments with the benchtop as well?
Goal. That's our goal, 2D experiments, 2D correlations, zero high field NMR correlations, and HSQC, HMBC type of information. So correlation between N15 and hydrogen. It's a goal. We are not there yet. It's not, I think, fundamentally impossible. It's just, again, practical challenges. Indeed, you want to be robust as practical problems associated with that. For example, of your solvent. I don't know, hopefully my internet is working. So one of the practical problems is evaporation of the solvent, and catalyst deactivation is as well. So we have this beautiful catalyst which binds both hydrogen and the substrate, but in fact, there is disactivation. We know that over time, even if the solvent stays on a good level, we still see decrease of the signal with time. So some of this chemistry we think we may understand, some of it we still don't understand, and I think it's very also system specific, depending on certain ligands. You have one chemistry, and with other ligands you may have other deactivation processes. So I think it's very very interesting direction to do 2D spectroscopy with enhanced.
Yeah, very cool. Maybe I also missed something. I wasn't quite sure why this is not happening. So you showed that with low field, you're able to see mixing between protons as soon as you bubble the hydrogen in, and then you do the polarization transfer to heteronuclei with pulse sequences directly. Why is there also, do you also expect any heteronuclear mixing at these really low fields, at zero fields?
At zero fields, yes. So here, right, that's exactly what we tried. We only tried millitesla fields. So we haven't actually, so in our lab, we also work with zero field NMR. So that's where we also want to detect our signals. And indeed, the next step with this particular work is to go to zero field and detect spectroscopic features of urea at zero field without any necessary polarization transfer scheme. So because polarization transfer will happen naturally if you go in a regime where hydrogens and nitrogens are strongly coupled, which is going to be below about a few microtesla, actually, in this case.
I see.
So yeah, it's possible directly in microtesla. It's a way of mixing indeed heteronuclei and polarization transfer.
I see. I think the talk was so good that I think the audience probably does not have any questions so far. But I probably might have one or two more questions.
Okay.
And so I was also curious about, like, initially we're also talking about these like predominantly all these homogeneous catalysts. And you said something about the fact that maybe not too many heterogeneous catalysts have been explored. I remember seeing some work a long time ago from the Pines lab, where they were, I think, imaging some heterogeneous catalysts with parahydrogen. This was a science paper from like 10, 15 years ago, if I recall correctly. So like, are any of those types of catalysts like routinely used at all?
Yeah, I think so. First of all, I might talk once again, maybe people listening from different fields. So there's a lot of research going on in heterogeneous catalysis with parahydrogen. Right. So it's groups of Bowers, Clifford Bowers himself, Igor Koptyuk, Kirsten Muniman, many more, Gerd Bunkovsky, other researchers. So it's just my opinion. I would say it's not necessarily always going to be the case that homogeneous catalysts are the best. So in my opinion, just currently the leaders, we produce much larger polarization values. And yeah, so it's actually quite interesting what to do in order to obtain high polarization in heterogeneous systems. And there's a lot of questions, and there's a lot of actually immobilization strategies. And so what was demonstrated by Pines and Louis Bouchard and Igor Koptyuk also in 2007-8, there is a portfolio of works using gas phase reactions, using these solid supported nanoparticles as well as immobilized catalysts. And there's a few papers showing imaging. So I think this direction definitely continues, and it's not finalized in any way, in the sense that now, if you're really talking about applications, if you're talking about something beyond the simple, not always actually simple, even demonstration, now the question is, okay, what actual use can of it, right? If anybody would want to use this type of enhancement and processes for their systems. And that's, I think, there is a little gap. I don't think it's a large gap. It's probably a little gap between very interesting directions of research, like fuel cells, as I mentioned, hydrogen energy, nitrogen activation, actually nitrogen with hydrogen, and other different processes, which we just need to understand and put together kind of conceptually. What do we want to study and how we are going to do it? How are we going to use homogeneous catalysts? Do we need to filter out unreacted precursors? If it's biomedical applications, probably yes. If it's industrial applications, maybe not. So I think it's coming in the direction of engineering more than actually science now.
Got it. So we do have a question from an anonymous attendee. Can you comment on the special detector used in the at this field, like SQUID, or I think they're referring to magnetometer, SQUID, and NVs, like advantages or disadvantages and applications?
Yeah, I mean, I can comment basically. So this research and this talk in particular was centered around chemistry and how we produce this hyperpolarization. But now, if you ask next question, okay, we have this technology to produce large polarization, how we can detect it indeed with other types of sensors and what their use can be. So I think general atomic magnetometers are just the most sensitive and most convenient sensors right now. So I think SQUIDs so SQUID, it's quantum interference devices. They're allowed to measure very low magnetic fields, and they require superconducting conditions. So there's usually bulky devices, so you cannot really move them. And atomic magnetometers, or what I call here optically pumped magnetometers, OPMs, it's a different technology, which is based on vapor cells. So there's cell made of glass with some rubidium or potassium or cesium atoms evaporated, and in this gas phase. By using lasers, you can polarize electron spins, and they act as very sensitive sensors of magnetic fields. So these are most, I would say, among the most sensitive, the most convenient devices currently to use, because you can just now buy them. They're becoming more and more available, mainly for magnetoencephalography purposes, as well as magnetocardiography purposes. So measuring magnetic signals from living species, either from the brain or from the heart. And this is just an interesting tools to explore regimes like zero-field NMR. They're most sensitive, they're relatively easy to use, and they will sense your spin chemistry at the zero-field conditions. So what about NV centers? I think it's very, very attractive platform. And there's a work or a few works in this direction and combination with parahydrogen, but I think it definitely can be explored much further. And the advantage of NV centers, as far as I can see that, it's the fact that you can actually do everything on a very small scale. So with even squids and OPMs, we're still talking about few millimeter size samples, right? And even at lowest, but NV centers, we can go to level of micrometers, potentially, so we can bring our sensor very close to the sensing area. And maybe a little practical advantage of NV sensors, they have very broad bandwidth as sensors, a few kilohertz, actually, if I'm not mistaken, compared to like few hundreds of hertz for OPMs. But again, for chemistry in liquid state, I think there's no much advantage from the bandwidth point of view. It's rather like ability to go into really small samples, potentially things like individual cells. If you really can measure metabolism in this individual cell and you can see differences between cells, I think that's a really goal worth pursuing in this kind of domain.
Yeah, very cool. There's also a question from an anonymous attendee on, like, could you comment on the challenges in the field of parahydrogen-based MRI?
Oh, there's a lot of challenges, of course. So again, I think there's challenges. There are fundamental. We can always divide challenges into fundamental and practical challenges, right? So I would say that in the field of parahydrogen enhanced MRI, there's still a lot of practical challenges. So let's say you want to polarize a molecule for in vivo applications. And I think currently with parahydrogen, we are talking about few molecules which reached the point where they can be used in vivo, in a sense, even potentially in a clinical setting. So there's that clean, and these are fumarate, and potentially we're talking about pyruvate as well. So I don't know whether we can really compete at this moment with exact levels of polarization with the dissolution DNP. At the same time, we're not that far, and the technology to clean up solutions, again, it can be based on different principles. You can either precipitate your solution or use different solubility tricks with different solvents and salts. And I think it's just engineering. So you need to spend enough time as a team of good, experienced engineers to put things together. Other than that, you know, there's no fundamental challenges. And usually in science, when our goal as scientists to publish, to write papers, we're not always focusing on finalizing, let's say, the device. Although, for example, in my opinion, urea could be one of these tracers. And I'm not even saying that this presented methodology is the final methodology to polarize urea. It's certainly one of them. We can produce already more than 1% polarization. So I think it's worth optimizing, going towards more reactor setups where we do reactions in more actually violent conditions in terms of parahydrogen bubbling, for example, compared to what we do in NMR tubes. So it's all about engineering. So that's a challenge, and a main challenge, I think, engineering time, putting people together to get this working on the molecule that you want.